Assalamu alaikum. Beta, in this video lecture, I will deliver the lecture on chromosomal abnormalities. I'll break this lecture into two parts. Starting with this lecture on chromosomal abnormalities, you first need to have some idea about very important terms, starting with the karyotyping. What is this term karyotyping, beta? It is the study of the chromosomes. To look at the chromosomes, their number, their normal shape, the way they should be. Okay, so the karyotyping is to completely study the chromosomes. Okay, now the question is, how can we study the chromosomes? Uh, the usual procedure to examine the chromosomes is to arrest the dividing cell in metaphase stage of the mitosis. Okay, the question is, why can't we just simply study the chromosomes? We take a cell, get into the nucleus and study the chromosomes. Why it is not so simple? Why we first need to divide the cell which is undergoing metaphase stage of the mitosis? Why? Because in remaining all the stages, the cellular chromatin material, the chromosomes, they are in dispersed form. They are present like this, and this is very thin, so it can't be visualized. And the metaphase is the stage where this chromatin material gets very much condensed like this, and the chromosomes can be seen. Okay, and now you know why do we first need to arrest the dividing cell in the metaphase, to have the condensed chromosomes which can be visualized. However, if you want to perform karyotyping in other stages of the cell cycle like prophase, anaphase, telophase, you can't. Why? Because the chromatin is very much dispersed by that time, and this thin thread-like structure can't be visualized, obviously. So metaphase is the stage for you where the chromatin material is very much condensed and it can be visualized. But still, it is condensed, still you need to give some stain, some color, because chromosome is colorless body. Chromosome is colorless body. You need to give some stain, some color to have visualization. So first thing is to arrest the dividing cell in the metaphase stage. Okay, number one thing, arrest the cell. In what stage? Metaphase stage. Okay, and second thing is to color the chromosomes. Okay, that is staining of the chromosomes. We will use a stain, you will see later. Okay, then you can visualize it. So first thing, how will you arrest the cell in the metaphase? By providing a particular uh, compound in the form of the drug that will inhibit the mitotic spindle inhibitors. So that drug is N-diacetyl N-methylcolchicine. It's a mitotic spindle inhibitor. It will inhibit the mitotic spindle, so the chromosomes will remain in this metaphase stage. They will not progress to the next stage. Okay, so now you have arrested the cell in the metaphase stage by providing this drug. And the number two step is to color. Okay, you have arrested the chromosomes this way. Now stain them. Now provide some color. So that is called beta G bending. A variety of staining methods have been developed for identification of the individual chromosomes, by the way, but the most commonly used is gypsa staining. Okay, so GIMSA staining, it's actually the stain which can be used to color these chromosomes to have the better visualization. But why do we call it G bending? What does this mean by bending? Actually, this is due to the reason beta, because if let's suppose this is the chromosome arrested in the metaphase stage, and we provide this GIMSA stain at some places, like this is very much, it takes lots of color and it's lots of stain, and at some places it takes lots of color, and at some places it is lighter, at some places it is dark, at some places it is light, and again at some places it is dark, again light, again dark. Can you see these bands of dark and light areas, light area, dark area, light area, dark area? Because this GIMSA stain is not uniformly picked up by the chromosome. The Jimsa stain is picked up in the form of these bands. So this is why we call it G bending. Okay. And by the way, this is the most commonly used stain for the chromosome visualization in the routine practice. Okay. Which of the following? It is the G bending. Jimsa bending. Jimsa is the stain. Bending. Why do we call it bending? Because the chromosomes picks the stain in the form of bands. Okay. You clear with the term karyotyping, hopefully. Now, let's come to the type of chromosomes better. On the basis of the centromere, you can see very clearly that if the centromere is present right in the center, and this above two are the P arms and below two are the Q arms, if the both the arms are equal, this is called metacentric. And if this is a little bit unequal, centromere is a little, at the top, not really at the top, it's just above than this, you compare, and the P arms are little smaller than the Q arms. This is called submetacentric. And if it is very much unequal, that the P arms are very much short and the Q arms are very much long, okay, this is then called acrocentric. You have to keep these terms in your mind. And if the centromere is at the very proximal pole, very top end, this is then called telocentric. Okay, these are some types of chromosomes you have to keep in your mind beta. After this is some types of chromosomal mutations. This is a really important part beta of this lecture. For basic genetics, for clinical genetics, types of chromosomal mutations. It can be deletions, it can be addition, that is gain, or it can be translocation. If let's suppose this is the gene sequence, nucleotide A, B, C, D. If let's suppose this is the normal sequence of the gene, this is nucleotide A, B, C, D. Okay, this is nucleotide 1, nucleotide 2, nucleotide 3, nucleotide 4. अगर बेटा इसमें कुछ यूं हो जाए कि एक न्यूक्लियोटाइड फिर दूसरा फिर ये आ गया फिर ये आ गया एंड देन ये, व्हाट इज दिस कॉल्ड बेटा? This is obviously not normal. This is addition, that is gain. Or if it becomes like this, A, C, D, what is this beta? Only one nucleotide. There is a change of only one nucleotide, not a complete chromosome. This is deletion for sure. Okay. And translocation is when, let's suppose this is your chromosome and this is another chromosome. No drawing can be worse than this, but okay, you can still understand. This is chromosome, this is another chromosome. If let's suppose a particular gene from here shifts to another position at another locus in another chromosome, this is called translocation. Okay, I will better explain you all these three terms in the next few slides. Okay, I hope you are clear with these deletion, addition and the translocations. Now there is a concept of the aneuploidy and the polyploidy. Polyploidy, that one complete set of the chromosome is added. In human beings, what is the normal set of the chromosomes? That is 2n. Okay, n refers to haploid number of chromosomes, that are 23. That makes up your 46 chromosomes. Okay, if in this 46 chromosome one set of chromosome is added, that is polyploidy. Not only one or two or three or many. Okay, so I don't know what does it make up. Okay, it can be 69, I guess. Okay, so this is polyploidy. Or if like this, 46 plus 46, this is addition of the two sets of the chromosomes. This was the one set of the chromosome, here is the two set of the chromosome, so this will be 92. Okay, this is polyploidy, when one or more complete set of the chromosomes are added. This is one complete set, here is the addition of two complete sets. Okay, this is called polyploidy beta. I hope you are clear with this, polyploidy. Okay, if I now come to aneuploidy, aneuploidy is not related to the complete set की addition या deletion. It is related to change in number of chromosome, only single number of chromosome, one or two number of chromosomes. Okay. So for better understanding of aneuploidy, let me show you here. That okay, what is aneuploidy beta? Aneuploidy by definition, it is the number of chromosomes which are altered. एन्यूप्लॉइडी का क्या मतलब है बेटा? अल्टर्ड क्रोमोसोम नंबर्स. देर आर 4 types of aneuploidy which I'll explain you on the next slide. But what is the reason behind aneuploidy? Aneuploidy, why aneuploidy? What is aneuploidy? Alteration in number of chromosomes. What are the normal number of chromosomes? 46. Alteration can be like one added chromosome 47, it can be like 45 one removed chromosome, or it can be two added chromosomes like become 48. matlab it can be like two chromosomes deleted, it can be like 44. Okay, this is aneuploidy. The number of the normal chromosomes is altered. Number of the normal chromosomes is altered this way. Okay, so this is aneuploidy. Now on the very next slide I will explain you what is the cause behind aneuploidy and what are the types of the aneuploidy. To better understand the cause of aneuploidy, you first need to understand the normal meiosis. What happens in meiosis, if consider this is the main cell and if let's suppose इसके अंदर आप फर्ज करें बेटा कि दो क्रोमोसोम्स मौजूद हैं. This is chromosome number 1, this is chromosome number 2. Okay, two chromosomes are present, and what happens in meiosis 1, that the number of chromosomes are reduced to half. Number of chromosomes are reduced to half. One chromosome is received by one cell, another chromosome is received by another cell. This is what happens in normal meiosis 1, and then occurs the meiosis 2. What happens in meiosis 2? The simple mitosis repeats. सिंपल माइटोसिस रिपीट्स कि अब ये एक क्रोमोसोम है बेटा, एक क्रोमेटिड इसके पास आ जाएगा, एक क्रोमेटिड इसके पास आ जाएगा. एक क्रोमोसोम के अंदर दो क्रोमेटिड होते हैं, एक ये क्रोमेटिड एक ये क्रोमेटिड. एक क्रोमेटिड इसके पास आ गया, एक क्रोमेटिड इसके पास आ गया. इसी तरीके से इसके साथ भी होगा. एंड एट द एंड ऑफ द मियोसिस 2, एट द end of meiosis, this was the meiosis 1 by the way, okay, where the number of the chromosome became half. This is the meiosis 2 where the number of chromosome remains same, that is still there is. Okay, this cell was having one chromosome, this was having one chromosome. You see, at the end of the meiosis 2 the number of chromosomes are same but the cells are doubled. This is having one chromosome. It is not a chromosome by the way, it is a chromatid, but later when this cell will grow, this cell will go into the S phase of the cell cycle, it will develop its another chromatid. Okay? It will develop another chromatid in the S phase obviously, so it will become a complete chromosome. Okay, so are you clear with this normal meiosis? Okay, in the meiosis 1 the number of chromosomes become reduced to half, and in meiosis 2 the cells become double and the number of chromosomes remain same. Okay, if let's suppose beta, if there is any defect in the meiosis 1 or meiosis 2, that is called non disjunction. अगर meiosis 1 या meiosis 2 में कोई defect आ जाए, कोई अगर defect आ जाए तो फिर उसको हम कहते हैं non disjunction. Non disjunction, and non disjunction can lead to aneuploidy. Okay, can lead to aneuploidy. It can be anything like addition of one chromosome, deletion. Okay, so you will better understand. We'll be talking about the types of the aneuploidy. So what can be the defect at the level of the meiosis 1 and meiosis 2 that leads to aneuploidy, that is alteration in the chromosome number. बेटा meiosis 1 में क्या नॉन डिस्टिंक्शन हो सकता है, क्या डिफेक्ट हो सकता है? ये डिफेक्ट हो सकता है कि ये जो दो क्रोमोसोम है बेटा, ये इक्वली डिवाइड होने के बजाय कुछ यूँ हो जाए कि दोनों क्रोमोसोम एक ही सेल में रह जाएं, दोनों क्रोमोसोम एक ही सेल में रह जाएं और इस सेल को कुछ भी ना मिले. तो एक क्रोमोसोम के पास एक सेल के पास एक एक्स्ट्रा क्रोमोसोम आ गया और दूसरे सेल के पास no chromosome. Okay, so this will be showing extra number of chromosomes, this will be showing a lacking of a chromosome. Okay, so this is the defect that can occur at the level of meiosis 1 leading to aneuploidy. Okay, this is called non disjunction, where the chromosomes completely do not segregate. Okay, normally what should have happened, that one chromosome should have gone to this, one chromosome should have gone to this. But if there is non-disjunction, what will happen? Both the chromosomes will go into this cell and this cell will receive nothing. So this cell will come up, if this cell if progresses further, so the individual will come up with the cells having extra chromosome. This extra chromosome may be added to any chromosome, okay, 21, 22, 19, 18, okay, and this, if this cell progresses and develops the baby, and if this cell progresses and develops the baby, that baby will be having a lacking of one chromosome, okay, right. And okay, defect can also occur at the level of the meiosis too. Normally what happens that uh, one chromatid is divided to one chromatid is delivered to each cell. लेकिन अगर कुछ यूँ हो जाए कि बेटा अब मैं इसको यहाँ पे ड्रॉ करूँ अगर तो इजी रहेगा आई थिंक. जस्ट अ सेकेंड. Okay, just for understanding purpose. Okay, what was your normal meiosis? What was your normal meiosis, beta? Here, what was your normal meiosis? It was like this. Okay, this was a cell with two chromosomes. Okay, and what happened then in the meiosis 1? What happened in the meiosis 1? You developed two cells. Okay, one like this and one like this. This was normal. And then what happened? Two cells with one chromatid in each cell. Okay, one chromatid in each cell, one chromatid in each cell. I told you in the previous picture that what defect can occur at the level of the meiosis 1. ये हो सकता है कि दोनों क्रोमोसोम एक सेल में चले जाएं और एक सेल कुछ भी ना रिसीव करें. व्हाट प्रॉब्लम कैन अकर एट द लेवल ऑफ द मियोसिस 2? बेटा कुछ यूँ हो जाए कि देखें ये जो दो क्रोमेटिड हैं दोनों के दोनों इसमें आ जाएं और इसको कुछ भी ना मिले. सो दिस कैन बी द डिफेक्ट एट द लेवल ऑफ द मियोसिस 2. कि नॉर्मली ये होना चाहिए था कि एक क्रोमेटिड इसके पास और एक क्रोमेटिड इसके पास आना चाहिए. लेकिन अगर कुछ यूँ हो कि दोनों क्रोमेटिड इस सेल के पास आ जाएं और इसके पास ना आए तो दिस विल आल्सो लीड टू एन्यूप्लॉइडी. दैट दिस सेल इफ प्रोग्रेसेस फर्दर it will lead to the formation of the development of the baby with one extra chromosome. Okay, and if this progresses further and develops into the zygote, this will lead to the development of the baby with one chromosome lacking. Okay, so both are the types of the aneuploidy, with one extra chromosome and one lacking chromosome. Okay, because aneuploidy by definition is the alteration in the chromosome, either addition or lacking of the chromosome. So you saw the defect can be at the level of the meiosis 1 or at the level of the meiosis 2. So this defect is non-disjunction. Okay, this non-disjunction can lead to aneuploidy. What is aneuploidy? That is alteration in the number of chromosome. That can be extra chromosome or one lost chromosome or lacking of the chromosome, right? Other than non-disjunction, there can be something called anaphase lag. Anaphase lag. Okay, that can be the reason behind the non-disjunction. Normally, what happens that the cells are when at the level of the anaphase, they are completely separated chromatids. You can see they go towards the opposite poles. But if let's suppose one chromatid is lagging behind in the cytoplasm and it fails to segregate, it fails to separate, it fails to go to its pole, then it is called anaphase lag, and this can be the reason behind aneuploidy. Okay, this can be the reason behind एन्यूप्लॉइडी. जो ये सेल बनेगा उसके पास तो पूरे पूरे क्रोमेटिड होंगे, लेकिन जो ये सेल बनेगा इसके पास एक क्रोमोसोम लैकिंग होगा. सो दिस विल लीड टू द एन्यूप्लॉइडी. यू अंडरस्टूड द डिफरेंस बिटवीन बेटा नॉन डिस्जंक्शन एंड एनाफेस लैग? I never care beta. If the video goes a little longer, doesn't matter. But the thing is to deliver the proper concepts. You understood the difference between the non-disjunction and the anaphase lag. In case of non-disjunction, what was happening? One cell was having extra chromosome and one cell was lacking actually one chromosome. But in case of anaphase lag, you saw one cell is having normal chromosomes and one cell will be lacking chromosomes. So both will lead to the aneuploidy but in different fashion. Okay, so I hope you are clear with the concept of the non-disjunction and anaphase lag beta. These two are the big reasons, non-disjunction and anaphase lag, that will lead to aneuploidy. Okay, yeah. Now let's go a little ahead beta. There are four types of aneuploidy, not visible here, but I'll surely explain. It can be number one, nullisomy. It can be nullisomy. Yeah. So there are four types of aneuploidy, beta. One is the nullisomy. I have explained it here the better way. One is the nullisomy. One is the monosomy. And number three is the trisomy, and number four is tetrasomy. Okay, the normal number of the chromosomes are 46 in your body. If two chromosomes, if a pair of the homologous chromosomes is lost, okay, so that will become 44, and that is called nullisomy, right? Okay, if beta, this is the nullisomy, and if you know the normal number of chromosomes are 46, and instead of a pair of chromosome, only one single chromosome is lost. Only one single chromosome is lost, that is, it becomes from 46 to 45. It is called monosomy, okay, and it is called trisomy when one chromosome is added. 46 is the normal number of chromosome, and if one chromosome is added, that will become 47, and this is the trisomy, okay. And it is said to be tetrasomy when a pair of homologous chromosome is added. That is, it becomes, when pair is added, it will become 48, okay. Normal is 46, when pair is added, this is called tetrasomy, right? ओके, सो इफ दिस इज़ अ नॉर्मल होमोलोगस पेयर, तो अगर इसमें हम एक और पेयर ऐड कर देंगे टेट्रासोमी करने के लिए, तो इस पेयर के साथ एक और पेयर ऐड हो जाएगा, और बॉडी में बाकी जो आपके क्रोमोसोम हैं वो इस तरह पेयर होंगे, लेकिन एक जगह पे एक पेयर एक्स्ट्रा होगा, ठीक है। और अगर ट्राइसोमी की बात करें तो पेयर की जगह एक क्रोमोसोम एक्स्ट्रा होगा, कुछ इस तरीके से। अगर ये आप फ़र्ज़ करें एक क्रोमोसोम, ये दूसरा क्रोमोसोम और ये तीसरा क्रोमोसोम एक्स्ट्रा आ जाएगा, तो एक जगह पे पेयर की जगह तीन क्रोमोसोम्स होंगे, ओके। सो दिस इज़ ट्राइसोमी। पेयर लॉस्ट, नलिसोमी। वन क्रोमोसोम लॉस्ट, मोनोसोमी। वन क्रोमोसोम एडेड, ट्राइसोमी। पेयर लॉस्ट, tetrasomy, okay. Now let's come to another term, that is mosaicism. Mosaicism refers to beta two or more population of cells within the different chromosome with the different chromosomal complement in the same individual. बेटा इसको बहुत आसान तरीके से कुछ ऐसे समझाया जा सकता है कि मोसेसिज्म का मतलब होता है बेटा कि बॉडी में दो तरह के सेल्स का प्रेजेंट होना, कुछ इस तरीके से कि फ़र्ज़ करें आपकी बॉडी में 100 सेल्स हैं, फ़र्ज़ करें आपकी बॉडी में 100 सेल्स हैं, उसमें से कुछ सेल्स अफेक्टेड हैं, यानी कि उनके अंदर कुछ क्रोमोसोमल एबनॉर्मेलिटी है, और कुछ सेल्स नॉर्मल हैं। रिपीट इट विथ मी, व्हाट डिड आई से? फ्यू सेल्स आर हैविंग क्रोमोसोमल एबनॉर्मेलिटी एंड आउट ऑफ 100 फ्यू सेल्स आर नॉर्मल, ओके। फ्यू सेल्स आर हैविंग क्रोमोसोमल एबनॉर्मेलिटी एंड फ्यू सेल्स आर नॉर्मल। अच्छा, ये कुछ इस तरीके से हो सकता है कि बेटा 100 में से ऐसा हो सकता है कि 20 सेल्स जो हैं वो एबनॉर्मल हों और 80 सेल्स नॉर्मल हों, एक तो ये हो सकता है, ठीक है। और दूसरा ये हो सकता है कि 100 सेल्स अगर आपकी बॉडी में हैं तो उसमें से 50 के अंदर क्रोमोसोमल एबनॉर्मेलिटी हो और 50 के अंदर एबनॉर्मेलिटी ना हो, वो नॉर्मल हो। और तीसरी कंडीशन ये हो सकती है बेटा कि 100 सेल हैं आपकी बॉडी में, उसमें से 90 के अंदर क्रोमोसोमल एबनॉर्मेलिटी हो और 10 के अंदर एबनॉर्मेलिटी ना हो, वो नॉर्मल हो, ठीक है। तो बेटा ये हो जाएगा आपका लो ग्रेड मोसेसिज्म। लो ग्रेड मोसेसिज्म, दिस इज़ 50% मोसेसिज्म। दिस इज़ हाई ग्रेड मोसेसिज्म। वाई हाई ग्रेड मोसेसिज्म? Because the abnormality is very much high, okay. So this is mosaicism. Beta, if this thing is occurring in your somatic cells of the body, it is called somatic mosaicism. And if it is occurring in the gonadal cells, that are the testes or the ovaries, that is called gonadal mosaicism, okay. So a very good example is the trisomy 21, okay. What happens in beta trisomy 21? If this is the 21 chromosome pair, this is 21 chromosome pair, this is the 21 chromosome pair, and if one chromosome is added extra, that is trisomy 21, and it's very good example. And this trisomy 21, that is the 21 chromosome पे एक extra chromosome का होना, this will lead to Down syndrome, okay. So this addition of one extra chromosome on chromosome number 21, that is called trisomy 21. This is one chromosomal abnormality. So this abnormality can be seen in all the cells of your body. one of this condition can happen, that okay, if it can be happen like 20 say 20% of the cells of your body may show this condition, and the 80% cells do not show this condition. 80% cells are normal and 20% cells may show the abnormality. This abnormality, that will be called as low grade mosaicism. Okay, and it can be happen that 50% of the cells in your body may show this trisomy, chromosomal abnormality, and 50% cells do not show. And it can be happen that 90% of the cells in your body may show this chromosomal abnormality. That means they are having extra chromosome on their chromosome number 21. Or it can happen that the 10% cells are normal, so this will be the high grade trisomy 21. Okay, so trisomy 21 mosaic विथ वेरिएबल एक्सप्रेशन ऑफ द डाउन सिंड्रोम. अगर ये सिनारियो है जिसमें 20% सेल्स जो है उसके अंदर एबनॉर्मैलिटी है, 80% नॉर्मल है, तो बेटा डाउन सिंड्रोम के फीचर्स इतने ज़्यादा नहीं आएंगे. और अगर 100 में से 50 एबनॉर्मल हैं और 50 नॉर्मल है, 50 के अंदर ट्राइसोमी 21 है, 50 के अंदर नहीं है, तो बेटा इसमें भी एबनॉर्मलिटीज़ आएंगी, लेकिन इसके मुकाबले में ज़्यादा होंगी. ठीक है, एबनॉर्मलिटीज़ जो डाउन सिंड्रोम की जो फीचर्स होंगे वो इस केस में भी अपीयर होंगे, लेकिन एज़ कम्पेयर टू दिस इसके अंदर फीचर्स ज़्यादा होंगे डाउन सिंड्रोम के, क्योंकि जो एबनॉर्मल सेल्स की मिकदार है वो ज़्यादा है. एंड सेम इज़ द केस फॉर दिस बेटा. ओके, आई होप यू आर क्लियर विद दिस कॉन्सेप्ट ऑफ मोसेसिज्म. ओके, and then is beta, yeah, then is chromosomal deletions. Chromosomal deletion is very simple term, that is the loss of the segment of the chromosome. Okay, it can be the interstitial or it can be terminal deletion. Interstitial deletion, when the segment is lost anywhere between the chromosome. This is the one terminal of the chromosome, this is another terminal of the chromosome. If the chromosomal segment is lost anywhere between two terminals, this is called interstitial deletion. Look, this segment of the chromosome is lost, this is one terminal, this is another terminal. Okay, and the segment is lost between two terminals, this is called interstitial deletion, and this is the most common, by the way. Okay, and terminal deletion, when the chromosomal segment is lost from one terminal end of the chromosome. Okay, one terminal end, this is one terminal, this is another terminal. If the deletion occurs from the terminal end, it is called terminal deletion, and it is not common, it is rare. Interstitial type is more common. Now after deletion, let's come to beta duplication and then translocation. Okay, duplication is very simple, that means the addition of the chromosomal segment. This is the normal chromosomal segment which is followed by A, B, C, D, E, F, G. Okay, two types of duplication are there beta, tandem duplication and the non-tandem duplication. It is said to be the tandem duplication when the extra segment is added adjacent to the normal copy. This is the normal copy BC and the extra chromosomal segment is added just adjacent to it. Okay, and if the extra segment is added dispersed away from the previous normal segment, this is the previous normal segment, this is the normal segment, and the new segment is added dispersed away from the normal previous segment, this is called non-tandem or dispersed duplication. The tandem duplication, that means the segment is added adjacent to the previous normally present segment. Okay, and beta, if the newly added segment is present in the same order, that is B then C, this is called same order tandem duplication. And if it is present in the reverse order, that is first is C and then is the B, the first is the C and then is the B, this is called reverse order tandem duplication. Why do we call it reverse order? Because the arrangement is reversed. And why do we call it tandem? Because it is present just adjacent to this normal one. And same is the case for the non-tandem duplication. It is called same order when present in the same order, and if it is reversed, that is first C and then B, it is called reverse order non-tandem duplication. I hope you are clear with this term of duplication beta. So firstly I told you about deletions, its types, then duplication, its types. Now is the translocation beta. Now is the translocation. Simply translocation is the transfer of the segment of the one chromosome to another chromosome. It's of three types beta. One is the reciprocal translocation, one is non-reciprocal, and one is Robertsonian translocation. This is the reciprocal translocation. Let me zoom into this and explain you what I want to say. Just a second. Yeah. Reciprocal translocation is very simple, beta. That if look, this is the segment of the chromosome and this segment of the chromosome. These are the two different chromosomes. These are the two different chromosomes, beta. This segment from this chromosome and this segment from this chromosome, they two are shared. Look, this chromosome, this segment is removed and added here. You can see removed from here and added here, and this segment removed from here and added here. You can see this is the segment which came from here, and this is the segment which came from here. Okay, this is the reciprocal translocation. Simple give and take. Simple give and take. Both chromosomes shared with each other. Yeah. And then is the non-reciprocal translocation. Let's go a little ahead. नॉन रेसिप्रोकल ट्रांसलोकेशन, जिसमें क्या होता है बेटा कि फ़र्ज़ करें आपके अगर ये एक क्रोमोज़ोम है, ठीक है, और एक और क्रोमोज़ोम ज़रा बना लेते हैं, ये रहा एक और क्रोमोज़ोम. ओके, दीज आर द टू क्रोमोज़ोम्स. अगर बेटा नॉन रेसिप्रोकल ट्रांसलोकेशन कुछ इस तरीके से होगी कि इसमें से ये एक सेगमेंट निकल जाए और इसमें जाके फिट हो जाए और इसका कोई सेगमेंट निकल के यहाँ नहीं आए. आप इसमें देख लें बेटा. लुक, दिस सेगमेंट removed from this chromosome and added into this, and no segment from this removed and added into this. You can see this chromosome is this portion is removed from here and added into this. You can see this and this got larger because a segment is added, and this because this segment is removed, so this got smaller. Okay, this is the non-reciprocal translocation because two chromosomes are not reciprocating with each other. Not reciprocating with each other. Just one chromosome lost its segment and added into another chromosome, but another chromosome is not ready to lose its segment and add into this. In the previous one, two chromosomes were reciprocating with each other, were sharing with each other. अपना सेगमेंट निकाल के दूसरे को दे दिया और दूसरे ने अपना सेगमेंट निकाल के पहले वाले को दे दिया. So they were reciprocating with each other, and in this case they are not reciprocating with each other. Okay, are you clear with this concept of the reciprocal and non-reciprocal translocation, beta? Yeah, and now is the Robertsonian translocation. This is the translocation between two acrocentric chromosomes. Between two acrocentric chromosomes. These are the two acrocentric chromosomes. You know, I hope beta you remember the acrocentric chromosomes, जिसमें एक आर्म बहुत बड़ी होती है और एक आर्म बहुत छोटी होती है. You can see these are the two chromosomes with one very large arm and one very small arm. Okay, you can see what happened. Yeah. If I draw the kind of here, this is acrocentric chromosome with one very large and with one very small arm. Okay, one very large and one very small arm. These two are the acrocentric chromosomes. Okay, for better understanding I can draw it with different colors beta. If you see that this is a very short arm, here is the centromere. Okay, this is very long arm. Okay, and this is a centromere, a very short arm and a very long arm. Okay, this is the acrocentric chromosome. Acrocentric chromosome, yeah. Beta, the Robertsonian translocation or the centric fusion occurs the translocation between two acrocentric chromosomes, and typically the breaks occur close to the centromere. The break occur close to the centromere. If this is the centromere, from here the break will occur, and in this case, if the centromere is the break will occur. Break will occur right from here. Okay, close to the centromere, and these two will be fused with each other. These two will be fused with each other. Okay, if I draw it for you here, this is the centromere and this is a long arm of this part and a little portion which was left after this cut, and this part will be added over here. This part will be added over here. Okay, and this part and this part will be fused. So I can draw it like this part first, okay, at its place, and a little from here, and the left is here, and this portion here. So what happened? So the break is actually near the centromere in both the cases, and resulting in the formation of the one very large and one very small chromosome, okay. And this may be seen in case of the Down syndrome, that is trisomy 21. What happened? Transfer of the segments then leads to one very large chromosome and one extremely small one. This is the Robertsonian translocation, but I hope you are clear with this concept, okay. And after this, I'll tell you about the isochromosome, a very important term to understand, and the ring chromosome, okay. One is the beta ring chromosome and then the isochromosome. Ring chromosome occurs when, produced when a break occurs at both the ends of the chromosome with the fusion of the damaged ends. Okay. अब मैं इसको कुछ इस तरीके से समझा सकता हूँ बेटा कि अगर आप फर्ज करें ये एक क्रोमोसोम का ये सेंट्रोमेयर है बीच में, ये उसकी एक आर्म हो गई और ये उसकी दूसरी आर्म हो गई, ठीक है। और कुछ यूँ हो कि इसके जो टर्मिनल एंड्स हैं वो कट हो जाएं और बाकी जो ये पोर्शन है दोनों का ये आपस में फ्यूज़ होके एक रिंग बना ले, कुछ इस तरीके से कि बेटा ये आ गया आपका सेंट्रोमेयर और ये एक पोर्शन आ गया ये वाला और दूसरा ये पोर्शन आ गया, तो सम हाउ इट बिकेम रिंग, ओके, दिस इज रिंग क्रोमोसोम। लेकिन इसमें एक मसला है, ये बैक्टीरिया वगैरह में देखने को मिलता है, लेकिन इसमें कुछ पोर्शन जो है ये क्रोमोसोम का लॉस होता है। अगर ये लॉस ज़्यादा हुआ तो फिनोटाइप पे असर पड़ेगा ज़ाहिर है, लेकिन अगर कम हुआ तो इतना फ़र्क नहीं आएगा। इफ़ सिग्निफिकेंट जेनेटिक मटेरियल इज लॉस्ट, phenotypic abnormality will surely result, okay. But if this small segment is lost, there will be no such problem, okay. After this ring chromosome is beta another term, that is isochromosome. Again, very important and very interesting term to understand. Isochromosome, what happens in this is beta, normally what happens if this is a centromere and these are the arms, you know, normally what happens and when your cell is dividing, normally what happens that there is vertical division, कुछ इस तरीके से डिवाइड होता है बेटा, और क्या होता है कि दो क्रोमेटिड्स बन जाते हैं, ओके, दो क्रोमेटिड्स बन जाते हैं कुछ इस तरीके से और कुछ इस तरीके से, ठीक है। नॉर्मली दिस इज व्हाट हैपेंस, देयर इज द वर्टिकल डिवीजन and the one chromatid on this side and one chromatid on this side, okay. But let me tell you what happens that will lead to isochromosome अगर इसमें बेटा वर्टिकल की जगह पे कुछ जैसे हॉरिजॉन्टल डिवीजन हो जाए। If let's suppose this is arm A, this is arm A, this is arm B, this is arm B. Normally when there is vertical division, a chromatid receives arm A and arm B. This chromatid also receives arm A and arm B, okay. But in isochromosome, what happens? There is not the vertical division. If due to horizontal division, if there is horizontal division, what will happen, beta? What will happen if there is horizontal division? There is horizontal division, then it will be like this. Yeah, two arms there and two arms here. Okay, when there is horizontal division, so two arms like this, receiving both the arms which are similar, receiving both the arms which are similar. Normally when there was a vertical division, एक क्रोमेटिड के अंदर दोनों आर्म्स आ रही थी, ए एंड बी, ए एंड बी, तो फिर बाद में जाके ये क्रोमेटिड मल्टीप्लाई करता था तो यहाँ पे ए बनती थी, यहाँ पे बी बनती थी, सिमिलरली इसमें भी ए बनती थी, बी बनती थी यहाँ पे नॉर्मली। लेकिन अगर यहाँ पे हॉरिजॉन्टल डिवीजन हो जाए तो एक क्रोमेटिड के पास दोनों ए आर्म्स आ जाएंगे, दूसरे क्रोमेटिड के पास दोनों बी आर्म्स आ जाएंगे, तो जेनेटिक मटेरियल सेम हो जाएगा, ठीक है। दिस इज सम प्रॉब्लम। तो फिर ये बेटा क्रोमेटिड जब आगे इंटरफेस में जाएगा तो वहाँ पे जाके क्या करेगा, ये ए आर्म है, ये दूसरी ए आर्म है, ये ए आर्म आगे क्या बनाएगी, दूसरी ए आर्म बनाएगी, ये ए आर्म क्या करेगी, दूसरी ए आर्म बनाएगी। द सेम इज द केस फॉर दिस क्रोमेटिड। सो नॉर्मली अगर होता तो ये यहाँ से ए आर्म बनती, यहाँ से बी आर्म बनती, एंड इट वाज अ नॉर्मल हैप्पी लाइफ। बट इफ देयर इज ड्यू टू सम रीजन देयर इज हॉरिजॉन्टल डिवीजन, तो एक क्रोमोसोम के अंदर सारा जेनेटिक मटेरियल सेम हो जाएगा। ओके, आर यू क्लियर विद दिस? आइसोक्रोमोसोम, आइसोक्रोमोसोम फॉर्मेशन रिजल्ट्स व्हेन वन आर्म ऑफ अ क्रोमोसोम इज लॉस्ट and the remaining arm is duplicated. So remaining arm is duplicated, only single, resulting in the chromosome consisting of two short arms only or two long arms only. You can see only two short arms or two long arms. Okay, either two short arms will be multiplying or two long arms will be multiplying. But normally what was happening? Both were multiplying. A, that was the short arm, B, that was the long arm. Both were growing. Okay, but if the isochromosome occur, what will happen? Only either A arm will be growing, either B will be growing. And that's all about beta this lecture. I'll continue this in the part 2 of the video. Will be describing the chromosomal abnormalities. Thank you so much beta. Class dismissed.